Okay, go ahead and come on in, guys. We got an exciting lineup of featured talks from the contributed orals in this second session of the afternoon. And don't feel shy, you can come sit up front. You know, there's lots of space up here. You'll be close to a microphone in case you have questions. If you have colleagues still in the foyer, feel free to tell them to come on inside. All right, well, as people are filtering in, I would like to just introduce myself. I'm Erica. This is Laurence. We're your moderators for this last session of day one. We have 8 lovely and diverse talks from the contributed orals. The talks are 15-minute slots, so 10 minutes for a talk, a few minutes for questions, a few minutes for transition. Same as we just had in the plenaries. If you have questions, you'll ask them into the microphone, and Laurence and I will moderate. Again, feel free to sit up front. This is a real big room for not a lot of us. And with that, I think I will introduce Siobhan Leachman, who is our first speaker for the afternoon. Take it away, Siobhan.
Kia ora koutou katoa. As I've just been explained, I'm Siobhan Leachman. I'm actually a Wikimedian from Wellington, New Zealand. I was Te Papa's Wikimedian in residence and undertook Wiki Project Te Papa research expeditions with my co-author, Lucy Schrader, who's Te Papa's Digital Outreach Manager. Now, the QR code on the slide, along with the bit.ly link, will take you to our slide deck. This contains links that may be of interest. The QR code will be repeated at the end of the presentation, so don't worry if you miss it now. So this is an overview of what I'm going to be presenting on. I'll explain about the project, the outcomes, and also the reuse of identifiers and data created by the project. I'll touch on a data sovereignty issue and give you a link where you can find much more information about the project. So the Te Papa Research Expeditions project was part of an ongoing international effort to create a global knowledge graph for research expeditions. We use Wikidata as our repository for our data. The umbrella project for this effort is called Wiki Project Research Expeditions, and it's associated with a TDWG Modelling Research Expeditions task group. Sabine von Mering is actually going to be giving a presentation on that task group later in this conference. So what is Wikidata? It's a sister project to Wikipedia. It's a free, open, structured knowledge base. It's multilingual by design. It can be read and edited by both humans and machines. It has a large volunteer community who are constantly enriching the data. All the data is licensed under a Creative Commons Zero license, meaning it can be reused without restriction. Importantly, institution identifiers can be added to Wikidata. So Wikidata acts as a global identifier hub, linking one institution's knowledge on a subject to another institution's knowledge. Wikidata is also queryable, for example, through its query service or its API. And this structure, openness and accessibility makes Wikidata a really powerful way to collate data and also makes it able to be queried, the data to be queried, so it can be used to answer really complex questions. So the Te Papa Research Expeditions project was one of several testing the international group's data schema against real-world data. Our project aimed to add data about Te Papa-related research expeditions to Wikidata using the proposed way of structuring that data. Any issues discovered during our project were documented to help improve both the schema itself and also the best practice guidance, which is currently being developed. And this project was also a practical test of how a natural history institution can use the schema. How does a place like Te Papa collate, translate and publish data about their expeditions? And then how can Te Papa actually make use of this work? Lastly, we aim to undertake outreach with both Te Papa staff and members of the public, educating them about Wikidata, building wiki editing skills, and empowering both these groups to add their own contributions about this topic to Wikidata, to the image repository Wikimedia Commons, and to Wikipedia. So here's our wiki project page for Te Papa Research Expeditions, and this is where you can find quite a lot of information on our project. Now, like many natural history institutions, Te Papa lacked a comprehensive data set of all their research expeditions. So a lot of my work at the start of this project was undertaking research. Most data about Te Papa expeditions was spread across a variety of sources, and these included publications, reports, archive records, even specimen labels. I then manually created new Wikidata items for each research expedition and added data to those items following the research expedition schema proposed by the international collaboration and shown on this slide. And I aimed to support each statement I added to each Wikidata item with references, making it clear where I actually got the information from. So we created about 120 research expedition items, and then also created or enriched many more items relating to expedition participants, publications created, locations visited, vessels used, taxa discovered, holotype specimens collected, just to name a few. And in doing this work, our project actually confirmed that the research expedition schema worked really well. We gave feedback on the schema to both the International Wiki Project and also to the TDWG task group, empowering these groups to make some improvements to it. Other outcomes included images being published from Te Papa's collection online website into Wikimedia Commons, and the creation of multiple stub Wikipedia articles for staff and the public to work on during the 2 research expedition Wikipedia editathons that we held. But more importantly, Te Papa then took this work and then built on it. Te Papa was interested in using the data generated from the project and has started creating records for each expedition researched in their collection management system, EMU. They round-tripped the data between EMU and Wikidata, and they did this by recording the Wikidata item identifier, also known as the Wikidata QID, on their own records, and then adding Te Papa's identifier for that research expedition into Wikidata on the research expedition item using Te Papa's agent ID property. Te Papa also helped improve Wikipedia. My co-author Lucy designed a Wikipedia article info box template for research expeditions, and this template can be placed on any English Wikipedia article about research expeditions and pulls appropriately referenced data from Wikidata and then displays it on the article, and you can see an example of the template up there. Te Papa also hosted 2 successful Wikipedia editathons, and this resulted in the upskilling of staff and the engagement with the public, as well as improvements to multiple articles relating to Te Papa's research expeditions. Now, I just want to go into a little more detail about the mapping Te Papa used to create research expedition records in EMU. These records have been created using a partial mapping of the research expedition schema, taking selected values and applying them to EMU fields. Now, Te Papa decided to do this with only a subset of terms because a full mapping would have taken a lot of modelling and development work. Full mapping was also seen as unnecessary because their EMU record was being linked to the appropriate Wikidata item for the research expedition that they were dealing with. Te Papa staff were able to access all the collated research expedition data in Wikidata just by clicking that link. So the new expedition records in EMU are party records, which empowers the research expedition to be added to collection event records as a collector, and from there can be linked to individual specimen records. The expedition record then gets published to Te Papa's Collections API, and from there it shows up on their Collections online website. So here you can see screenshots of Te Papa's Collection online website, which shows a Te Papa related research expedition, the specimens collected during that expedition, and the Wikidata item associated with it. There's also a screenshot of the identifier section of that Wikidata item, showing the Te Papa agent ID has been added to it. And with these connections in place, Te Papa's now looking at what else can be done, such as using the Wikidata QID as the parent event identifier when publishing occurrence data into GBIF. Now, I also want to address a data sovereignty issue that resulted from this project. Like museums, research expeditions have been entwined with colonialism for centuries. Work during projects like ours is likely to find information that's relevant to Indigenous communities. Now, our project had one historical expedition that was intimately tied to the source community's knowledge about medicinal plant use, and we were very aware that this Indigenous knowledge would become much more easily findable and reusable if the expedition data was placed in Wikidata. So discussions were held with Te Papa's Matauranga Māori team and provided guidance that these cases need to be worked through with the relevant Indigenous community, following Te Papa's principle of mana taonga, as well as the care principles of Indigenous data governance and the Māori data governance model. Now, we go into a lot more detail about this in our report, which we produced at the conclusion of the project. This report also gives much more detail on the project achievements and outcomes upon which I'm presenting today. And I believe that we actually showed this is a really valuable project for a natural history institution to undertake. It surfaced knowledge previously spread across multiple sources and made it immediately reusable. It created opportunities to enhance data in the institution's collection management system, as well as in Wikidata. And it supported outreach to the public, as well as others in the GLAM sector. Plus, Lucy and I found it really fun. So if you want to find out more, talk to me during the conference. You can also contact Lucy at Te Papa, check the project page on Wikidata, or you can read the final report. Thank you, and does anyone have any questions?
Thank you so much, Siobhan. And if you do have questions, please find your way to one of the microphones around the room. You might need to turn it on. While the room is thinking about questions, I do have one for you. I'm really curious, this is a very interesting project, and I'm curious if you can talk about the balance between where to store canonical information. So is Wikipedia the source for like the most authoritative version of a research bit of information, or is EMU, or does that differ depending on what the information is?
I think it depends on what the information is. Obviously, I don't think there is a place where I would like to think that there would be a wonderful, detailed Wikipedia article in multiple languages that anyone can access about each particular research expedition. But not every research expedition is notable for the purpose of Wikipedia. In that case, I would like to think there would eventually be a Wikidata item that is chock full of data about that research expedition. But again, it's possible that, and I have come across expeditions where I just cannot find who the participants were. I would like also to think that if there is more data in a collection management system, that collection management system is accessible to people who want to get access to that data. So I relied a lot on Te Papa's Collections Online version, because I knew that information was public. I refused to actually look at EMU, because then I would have to worry as a person putting this information out into the public about privacy concerns, about Indigenous knowledge concerns. And so for me, I accessed the information on their actual public online website rather than looking at their EMU record for each expedition that they had there. But they had 50, or I think it was 48 expeditions in their EMU when I started. Now they've got over 120, yeah. So I've doubled it. But this was only a pilot project. It was only 12 weeks. I'd love it if someone, did a lot more work on their expeditions, because it's untapped resource of a whole load of biodiversity information.
Very cool. Well, Siobhan will be around all week if you have questions for her, and I'd like to give her another round of applause. Thank you so much. Our next speaker is Arturo Ariño, and we'd welcome him to the stage now.
Thank you. Well, I'd like to thank the organization for the session, and thank you all for coming all the way here despite that little drizzle over the center of Japan. Anyway, let me start by misquoting one of my favorite authors. Yes, we do important things, or rather our forebears, many of which are here in this room, created a song that in theory is sung by many other scientists, technicians, managers. The stream of biodiversity data that feeds many endeavors is something for which we provided many necessary nuts and bolts, and such as data, data stewardship, and information access to everyone. So what do we do know what we do, and we regularly try to communicate to others, at least within our community, what we do, mainly through conferences, of which this might be the 40th or something in that week, I don't remember really. But the question is rather, do the others know what we do? Are we known? Do they use what we do? Well, what is mainstream? According to the dictionary, activity or ideas regarded as the most typical, normal, and conventional about nature and behavior, natural things. This is normally known through publication, and there is a conventional bitten of this, which is peer reviewing and indexing. So we might assess what is the mainstreamliness of our activity by measuring how it is formally published, how it is formally exposed through preferably indexed literature, and whether our works is showcased in indexed literature through citation levels, etc. The funny thing is that we can publish in non-indexed venues, and this might also be interesting or important too. So, okay, we have a product we want to show, and normally we will produce a paper, and there are two types of papers, as everybody knows. There are indexed papers and non-indexed papers. Let's say we have main literature and fringe literature, okay? But in practical terms, index means that the paper has been picked by a company which has looked at certain sections of the papers and has decided that this paper is important, so this journal actually is important, and other journals are not so important. Whatever the production, it can be cited by citing literature, which in turn can be indexed or not. And there are links that can be established by these indexing companies like Climate, Web of Science, or by Scopus, linking titles with literature and looking at that and counting how many citations you have, etc. But then we have Google Scholar, which is quite interesting because it could look at everywhere in the article, but it's automatic. It's not manually curated, which imposes a number of limitations, okay? So let's say that Google Scholar can look at fringe things and extract fringe citations that indexed companies cannot. And we can look for this literature through the main companies or through Google Scholar and actually find different results. But anyway, we can combine all of them into citation measures that try to measure this impact. So I measure the mainstreamliness through a sampling of literature, both located by indexed companies or by Google Scholar. Okay, I capture data, I look at several million records, and I take a sample of 100,000 and look at how do they cite each other and compare terms or searches pertaining to our remit with searches pertaining to others' remits, such as biodiversity, ecology, taxonomy, or even outgroups, such as, say, biomedical terms or artificial intelligence, which is not necessarily outgroup, but we might consider for the purpose of this outside. So those are the search terms I use. I use 19 separate sets of search terms divided into theme terms and comparison terms and outgroups. I did a lookup in Web of Science, in Scopus, in Google Scholar, and came up with a number of hits. So about 100 million terms, of which I started about 100,000 records to further analysis. I did some primary verification examples, such as looking for congruence between the searches. For instance, if something was searched in the title, probably it should appear also in the abstract as well. Well, basically the searches were consistent, and also, as we all know or suspected, of course, any search within Web of Science or Scopus only will retrieve a small subset of the entire thing. Google Scholar will retrieve most, but not all. There are some tiny regions of exclusivities of one of the other. But anyway, so after those quality checks, let's go to the results, which is what is important for you and for me. let's focus in four main outputs, or the outputs of four main things: spinach, tadwig, GBIF, and a specific, very specific output, which is Darwin core. Darwin core, which is the main product, at least for tadwig, which is basically the basis of everything else. The growth, this is a logarithmic scale. Don't be misled. The growth has been exponential in the case of GBIF, meaning that every 3 years or so, you have twice as many papers as 3 years before published from GBIF. The spinach or tadwig have reached a plateau, and they are relatively constant in terms of publications. And Darwin core is also appearing to increase, but not as fast as GBIF anyway. Well, the thing is that this is a search on Google search. So if I ask you how much of this production is actually indexed, do you think it will be lots of productions like in other fields or more than in other fields? How many of you put your hands up if you think that we'll have less indexed literature in proportion than in any other field? Very few. How many of you think that it's basically on the same league? Just a few. How many of you think that we are more indexed than non-indexed? In only one? Whoa. There's always somebody optimistic here. Okay. In terms of citations, we are not that far from other fields. We are basically in the middle, GBIF, or perhaps a little bit down, but this is indexed literature. This is indexed literature. Let's see what's the proportion. The proportion of was indexed literature is very, very small in the case of tadwig, spinach, GBIF output, Darwin core, or related concepts such as taxonomical database, biodiversity database, etc., as compared to other fields such as ecology, taxonomy. I have a specific one, which is species diversity models. And also, it doesn't change much from old literature to recent literature, okay? So, no, we are not indexed. We are fringe, in a sense. But let's have a look at a different thing. When a paper is published, it's cited later on, and this citation can be either indexed or not. So we can separate them if we use a Scopus feature, which is the possibility of looking at the references lists, which is a combination of both indexed and non-indexed literature. So we can construct a leverage of whether a production tends to be particularly appearing in indexed terms, in indexed literature, or tends to appear equally, or tends to appear most in unindexed literature. It turns out to be that tadwig and spinach have an enormous leverage towards indexed literature, which means that our production, even though it's not indexed, it ends up in indexed literature quite frequently, much more frequently than many other terms, especially in the general science terms, which occupy the lowest part of this plot there. We can confirm this, it's not that difficult, by looking at only within the index corpus of literature at whether spinach or tadwig or any of our production which is indexed appears in indexed literature. It turns out that papers feeding on our production, per paper providing this production, I mean indexed paper providing this production, is quite large. I mean that, for instance, for a product produced by tadwig, say Darwin core or whatever, to be picked in an index output, to be picked by another index output can be up to 4 to 8 times as likely as in most other fields, even related fields. Okay. In the case of GBIF, it's about twice as probable to have an indexed product from GBIF appearing in an indexed paper of any other field. So I would venture to say that we are not exactly fringe. We are fringe formally because governments, universities will only look at your index literature and say, no, you're fringe, you are not publishing in anywhere that is important, but the work we do is not fringe. That's what I think. Index research does use this kind of fringe index, unindexed output. So indexing is grossly misrepresenting the actual value in terms of mainstreamliness of the work we do here. Tadwig and spinach are carrying significant useful outputs. Another interesting thing is that when we search for the literature lists in Scopus, we can differentiate hits within the title or hits within the source. And as we know, we do have sources that tend to appear quite heavily. Everything that is above that diagonal means that the carrier, in this case, for instance, biodiversity informatics standards, or the outputs, the abstracts of these conferences tend to be overrepresented in citations, even though they might carry research which has little remit to spinach, tadwig, or GBIF. One last word. Even though our production tends to have a large proportion of output in abstracts or conferences, well, when we look at how it is cited, it's cited mainly in articles. And this also differs from other fields. Articles get cited by articles, conferences get cited by conferences. Tadwig, spinach, GBIF's conferences tend to be cited in articles. So, to end, I would say that we have a low index output, that's undeniable, but non-indexed output is cited very extensively, especially in the case of Tadwig, up to 8 times as frequently. And it is indexed literature that inordinately feeds on Tadwig, or spinach, or GBIF2 as well. Let's say that an indexed paper that containing terms or concepts related to Tadwig or GBIF remit might have an average of 50 citations, whereas in general science is 5. Therefore, Tadwig, I would suggest that Tadwig is actually mainstream, at least, spinach 2 and GBIF 2 to a different degrees, but the mainstreamliness of our activity should be probably recognized somehow. That's it. Thank you.
Thank you so much, Arturo. We don't have time for questions after this speaker, but you can find Arturo during the conference. And I'd like to welcome up Robyn Drinkwater as our next speaker.
Hi, so I'm Robyn from the Royal Botanic Garden at Edinburgh, and I'm going to talk to you about Frankenstein's plants, which is an event we developed to talk about herbaria to the public. So the herbarium in Edinburgh consists of about 3 million specimens, and the herbarium is based on the edge of the public gardens, but many people don't know what that mystery building is, so we needed to look at a way to address that. We looked at bringing it to the 2019 Edinburgh Science Festival, and that Science Festival has been running in the UK since 1989, and it's one of the biggest events in the UK, with around 90,000 people coming to it over a 2-week period in the Easter holiday, so this looked like the perfect opportunity for us. We developed it based on an idea we'd seen Q run, to start to look at how we could bring in the public to talk about herbaria with them. And the basic idea of the event is to go through the stages of creating a herbarium specimen. So when we were developing the event, we had a few key aims that we wanted to look at. One of them, say, is to raise the awareness of our herbarium in Edinburgh. The gardens themselves have around 1 million people coming to them to visit annually. And it's a really well-loved visitor attraction within the city. However, a lot of visitors to the gardens, local residents or international, don't know that we have a herbarium, and they don't know how that's used by scientists globally. We wanted to show people how specimens are made, how they are used, and what we can do with them once we've got them in our collections. And we wanted to introduce some basic concepts of what a scientific name is, how it's constructed, and how we can use that to communicate about plants and animals to people in research and why we don't want to be using common names when we're doing this communication. In addition to this, we were able to talk about some other STEM and science careers that people could have. You could look at curation, you could look at being a botanist. There's a whole world of other careers out there that people aren't always aware of, so this is an opportunity to start raising that awareness. We wanted it to be adaptable in both the material that we used and how it was staffed, so that we could run it either at the Science Festival or maybe at one of our regional gardens, so that we could take it out on tour. And we wanted to have an opportunity for staff and students in the gardens to do public engagement. It's not something we always get out to do, so this was a great opportunity to bring that in as something for people. The event itself is run in our main visitor centre, the John Hope Gateway, which is the sort of main point of access to the gardens, in one of the side rooms, and it's laid out with a series of stations where you go through the processes. And it's manned by herbarium staff and volunteers from the PhD and MSc cohorts. Being based in the visitor centre, we're able to pull people in as they're coming through and see if they want to have a go at making a specimen. And when it's quiet, we'll send people out to tout for trade and bring them in just to get people in. And at points we've had enough people, so many people in there that actually we're struggling to manage the capacity of the room we're in, looking at how we can add tables, looking at how we can add chairs to accommodate everybody who wants to be creating a specimen. We have quite a simple flow with several tables with posters behind explaining the different steps, and that's manned by staff at every point. And in the next few slides, I'll go through the different steps that we've got in our specimen creation process and how we can start to talk about the work of Herbaria as we're doing that. So the first step, which is selecting the material, is where we can introduce the concept of the event, which is to create a specimen or a creature, is what we're sort of generally saying you can do. And we're providing the participants with a selection of pressed and dried plant material that we've collected from the gardens or that we've pressed from flowers bought from online florists. So it's already ahead of the event. The first key step is to mount the specimen. So everybody's provided with a cut down herbarium sheet, so it's slightly smaller than A4. And we have pre-printed labels with some key collection information on them, such as locality, collector name, collector number, a barcode. And this is where we can start talking about the kinds of data we want to be collecting when we go out and bring specimens back for the collection, and how we might be using that data. People might ask us why locality is on there, and then we can explain how it can be used for research in the future. The specimens are laid out quite carefully, and this can be a really, not a slow process, an engrossing process where people are spending a lot of time arranging their material to create whatever it is they have in their imagination. And we're using gum tape to attach it because we found this to be the most low mess option we could come up with. We didn't want to have a lot of glue around because it would just create chaos for the cleanup. So this process is often, say, quite an engrossing step. People can take 20 minutes just to create whatever it is that they have in their imagination. So this is where we can start talking to either the participant or the family members of that participant about what a herbarium specimen is and why we go out and collect those specimens. The next step is to come up with a name for your creation. So we've got a selection of genera and species epithets that we've pulled together and put up on boards behind. We were saying you can do it based on your initials, so like your first name and your last name, so you could draw inspiration from that. But we're talking about the structure of a name and how we can communicate about plants with that. And some of the names we've got are real and some of them are made up. And it's sort of, I know we've got Darth Vader on there, so it's quite a nice one to talk to people about how you can have some humor or you can link it to something key to help raise awareness of a particular species. The next step is to describe the specimen. So this is where we can start to introduce some botanical terms, but this really does vary depending on the age of the participant. If they're particularly young, we might sort of pull that back and you might find that it's described as a happy flower. or that it's spiky. As the participants get older, we can start to introduce some of the botanical terms that we've got available on board, so that people can start to have a think about how to describe at least some part of the specimen that's there in front of them, and how we would use these terms when we're talking about a new species, for example. The final step is to digitize the specimen. So to digitize it, it's a very simple setup, and it's the same that we use in the herbarium. They scan the barcode and they click a button to take the photo. But it's a perfect opportunity for us to talk about how the staff at Edinburgh and at many natural history collections across the world are working together to digitize their collections to make them available to everybody, so that that information becomes much more globally accessible. We have a big screen connected to the laptop so that the images as they're taken come up for everybody to see. And this is often a really nice opportunity where people have a photo with their specimen, with their photo behind it, as a sort of memento of the event. And it seems to be very popular. We put it up to try and make it as a draw so people could see what was happening. And we weren't expecting people to want their photo with their specimen, especially as they get to take their specimen home. So it's something they do get to keep, but it is, say, a very popular part of the step. All of the specimens go onto an online herbarium, so we're using a Flickr gallery for this. It's free for up to about 1,000 images, so it's perfect for the event to run as it runs annually. And people are able to look up their specimens as there's a link to the site on the label, and there's a barcode on the specimen as well. So this is just a couple of examples. There's so many to choose from. I had a hard time picking some of my favorites from the selection. It's always been great to see what people come through. Having created, we don't have that much variety in the material we offer, so it's really fantastic to see what people can come up with based on what we've provided them, and they are all different and all really fun to see what people have been up to and what names they've come up with and how they've described them. So in terms of the event, it offers a lot of opportunities for us to engage with visitors. As I say, it can be quite an engrossing process. So often you've got parents or guardians waiting, and they're the perfect ones to start chatting to about what we do in the herbarium. We've got a small display about the collections in Edinburgh with some example
I could eat another
specimens.
couple of crumpets
There's a
from
nice
you,
captive
even
audience
if
to
they were
start chatting
bungles.
to. And it can be really fantastic conversations that we have. We find, because we're based in the Gateway, we're getting a high number of international visitors come through. So this is becoming part of their visitor experience to the gardens. And especially with the international group, we'll often find that the adults are as willing to take part as the children, whereas sometimes you get a lot more reluctance. And we do often see that maybe out of a group, not everybody wants to initially take part, but as they're seeing it happen, they're like, actually, I will have a go. So then they'll head off and get their own material and start making a specimen as well. In terms of local residents, at the same time of the Science Festival, we've also got the Easter Trail running. So there's a good attraction to bring children in to do the trail, win their chocolate bar, but also then get them in to make a herbarium specimen as well. And so far, the feedback's been really fantastic. It's been described as a great way to get children to engage with botany. It's creative, it's practical. And we've had a lot of informal feedback as well, where people are sort of saying, we've managed to get their teenager off their phone for more than 5 minutes, which is always nice to hear. And we've had a number of people asked to take photos of our panels and posters so that they can recreate the event either in a school or a group that they work with. And we're more than happy to share any information about this, share the posters we've made so that people can do something similar as well. We were inspired by Q, and we want to keep inspiring other people to talk about Herbaria and get people in and making their specimens. So over the 3 years that we've run the event, we've had around 1,000 specimens made, but we estimate, because of parents and guardians and other people in those groups who we're engaging with, that's maybe near 2,000 to 3,000 people that we've been able to talk to or show some glimpse of the herbarium to. And the age range is fantastic. We've got very little sort of youngsters taking part, up to sort of 93, 94-year-olds having a go. So there's no restriction on who can make a specimen. And we're looking forward to running it in the future. The plan is to take it to our regional gardens now. So we're just trying to work out how we do that. And we're hopefully running it again at the Science Festival next year. So thank you very much. And any questions?
We do have time for one question if there is one.
I know this was aimed at children, but did you get any volunteers for mounting specimens out of doing this?
Not directly, but I think we've had people asking about volunteer opportunities in general in the herbarium as well. So we have had that kind of interest from it. We've had a pause in our volunteer program during at least one of the years that we were running it, so we weren't able to take them, but we do get that kind of interest coming from it. So it's been good for kind of boosting that element of the work we do as well.
Thank you.
Thank you so much, Robyn. And our next speaker is Roderic Page.
Okay, firstly, apologies, I'm losing my voice. I've got some sort of slight cold. As you can see from the slide, I have enormous trouble deciding on titles. The title I gave is not really what I'm going to be talking about, but sort of, so apologies for that. I should also apologize, I'm going to speak very quickly. If you miss things, most of the slides will have a QR code, which will take you to the particular topic I'm talking about. I also have a blog, which is what that QR code there is for, which will help you sort of follow up some of the sort of things I'm going to talk about. And of course, I'm on the socials. I'm on Twitter, Blue Sky, and Mastodon. Quick question, how many people here are on Blue Sky? Oh, just a few. How many people here are on Mastodon? Maybe a little bit more. It's really interesting. My sense is that as the exodus sort of goes on, the phylogeny people have tended to go to Blue Sky, and the sort of open sciencey people more to Mastodon. It's just an interesting kind of time. Okay. What I'm going to talk about. This talk was partly inspired by another meeting. This is the Systematics Association. It's a nice little meeting in the UK. I was there this year. It's a nice small meeting with fantastic artwork that you can see here. And there was a symposium on the state and theory of the new taxonomy. And I went to that. And my God, it was a depressing experience. It was organized by Quentin Wheeler. Quentin has a really interesting podcast. If you want to listen as you quietly slash your wrists about the state of taxonomy, here's a podcast for you. Basically, he says taxonomy is in great trouble and there's a lot of blame to go around. Taxonomists have sold out the integrity of their own field. Institutions like museums and herbaria lack vision and have no backbone. And other colleagues, it's not just taxonomists, but everybody else, has been subject to greed, hubris, or they just do not understand taxonomy. So, if you don't want to sort of kill yourself by this stage, it's just extraordinary. But in the same meeting, pretty much the same symposium, we heard things like this. This is a project from the Natural History Museum in London, and they have been given £200 million to build that building and to digitize huge amounts of specimens. I mean, they literally have loads of money. So the question here is, what's really going on? What is the state of taxonomy? So how would we find this out? So one approach, of course, is we could ask taxonomists, if we dare. Now, there are projects that do this, like the Red List of Taxonomists is one. But I'm kind of lazy. Another way to do this would be, rather than ask taxonomists and bug them, we could try and see what they're doing. What are taxonomists actually doing? Now, the inspiration for this comes from a project that Dave Rimson set up probably nearly a decade ago. As many of you will be aware, Dave died a couple of years ago. This seems to be a really dangerous field to be in at the moment. We lost William Allanty last year. And those of you who are aware of the New Zealand sort of wiki and iNaturalist community and Taxacom, Stephen Thorpe, will be known to many people, and that sort of community was murdered horrifically about a week ago. So back to slightly less depressing things. So the reason I'm showing this picture of Dave is he and some other colleagues at Woods Hole set up a really cool project called UBioRSS. And the idea was really simple. They are going to find out what taxonomy is happening by basically using RSS feeds from journals, little sort of automated updates of tables of contents. They'll get those articles, extract taxonomic names, aggregate them together, and there's a project. It's a wonderful little thing. Sadly, like many projects in our field, it's gone now. And we've already had a few people sort of comment about this. We are a field full of really cool little projects that last maybe 5 or so years and they die. And that's a huge kind of problem. So I took this as inspiration and said, can we sort of bring this back to life? So I built yet another little project that'll probably last 5 years before it dies. There's a QR code there. This is called BioRSS. This basically does the same kind of thing that UBioRSS does, but with a few sort of bells and whistles. If you go to that website, you'll see something like this. So you've got, here are the species we're looking at, little tree map of the taxa. You can drill down, see if you like bugs or plants or fungi, you can have a look. It's filtered by country. Here we're showing China. And on the right-hand side, a list of all the papers that have come out in order which they appeared with some cute little pictures. Okay. So that's kind of fun. And by doing that, I got the sense there's a lot of taxonomy kind of happening. So I tried to sort of summarize that in some way. If you go to the website, you can see a page like this. This is for 2022. Left-hand side, where are most of these articles coming from? Dominated hugely by China, India, Brazil, Mexico, United States. On the right, what taxonomic groups are people studying? Lots of animals, mostly arthropods, lots of plants, and a few other things. So anecdotally, there seems to be a lot of taxonomy happening, and it's mostly in those countries, or maybe it's mostly about those countries. But if we want to really sort of drill down a bit, we want to do a bit more. So what I thought we could do is build, of course, a knowledge graph. You know, if I'm going to be talking, there's going to be a knowledge graph to connect people, publications, and funders to those species. Basically, where's this work being done, and who's paying for it? So I won't sort of go into the huge details of this. This is a very simple kind of knowledge graph. The key point here is I'm using two things. For any paper, if I have the ORCID ID of the author, I'm hoping most people here have an ORCID ID, ORCID will tell me who that person is, what works they've published. If they have a cross-ref DOI, I can also get information, particularly on funding, so what agency funded that work. And the one thing that's sort of lacking here and sort of hardest to get here, ironically, is the link between what I'm calling here the creative work, which is the publication, the article, and a taxonomic name. So the idea is we know who did the work, what the work was, who funded it. I want to find out from that. Now, I'm going to take 30 valuable seconds of my life to have a quick rant. It drives me utterly bananas that this particular thing here is the hard bit. It's the 21st century, and we do not have a comprehensive list of every taxonomic name on the planet linked to the publication that published it. So every time you hear a taxonomist complain about they are not being cited, give them a slap and ask the question, if you want to be cited, you need to have a database linking names to DOIs. Catalogue of Life, for example, less than 2% of the names in that database are connected to a DOI or any other identifier for those publications. Okay, back to normal service. So, given that we don't really have those sort of nice names linked to DOIs, I thought we'd use BioRSS to give us a subset to look at. And by we, I dragooned an undergraduate student, Ailey Richmond, to have a look at some of this information. So essentially, we got a set of articles from 2022. We know what part of the world they refer to. We know the species they refer to. Using Crossref and ORCID, we could find the funders. We use Wikidata, which Yvonne's already talked about, to find where in the world those funders are. And we could find out, okay, what countries fund taxonomy, what parts of the world. So I'm just going to give you some really quick, simple kind of results as maps. So this is a very common pattern. This is China. On the left, where does China fund taxonomic research? Surprise, surprise, China. Most countries do that. You fund taxonomy in your own country. But also you might fund it elsewhere, Southeast Asia, some of the United States. On the right-hand side is the taxonomy that is done in China. Where's that funding coming from? Again, no surprise, it comes from China. But other countries too contribute to taxonomy of organisms found there. This is the United States, which has a lot less taxonomy coming out of it, but has quite a sort of interesting kind of global spread. Lots of taxonomy all around the world is funded from the United States. Likewise, there's some taxonomy in America that's funded by other countries. We're in Japan. So I should do that. Japan has a very kind of typical kind of pattern. Most taxonomy in the country is funded by that country, but also research funded by Japan also includes regional kind of areas here. So Japanese people working in China, India, and so on, likewise, those kind of regions might similarly fund work in Japan. So this was kind of fun and kind of interesting, and obviously there's lots more scope to sort of drill down with this. Now, then it occurred to me, I got distracted, as I usually do. So we can get the funders and authors more or less for free from ORCID IDs and DOIs. What else can we get potentially for free to learn more about this, this network of people and publications? So I want to look at a project called Make Data Count. How many people have heard of this? Has anybody come across this? There's a few hands going up. I think this is potentially a really interesting project. This has been released recently by DataCite. So the idea is we're interested in tracking citations of data. Now, we're probably all very, very familiar with GBIF and the hashtag #CiteTheDOI. GBIF is very good at trying to track citations of whole data sets. What this is about is citing individual things, particularly, for example, DNA sequences, protein sequences, and so on. So they've just launched this, and they've started with 10 million citations, mostly of DNA sequences. So you imagine the ability to find out, here's a DNA sequence, who cited it. Now, sadly, of these 10 million citations, about half are complete bollocks. There's a blog post in iPhilo which explains why. This is one of those cases where AI seemed like a really good idea. The Chan Zuckerberg Initiative did this work, about half the citations are wrong, so it's a bit of a rocky start. But imagine extending this to having citations not just for sequences, but for museum specimens, herbarium specimens, and so on. This might be a project that we could use to sort of build that out. So I have an orange light flashing. Let me sort of summarize this. So what is the current state of taxonomy? I don't know. And if you thought I was going to answer that question, well, more fool you. I'd love to know. I'd love to have a platform that would actually help us do that. That actually tells for any tax in any country a year who is funding that research. But I think some of this is going to happen anyway. If we take ORCID and Crossref, they're pumping out lots of the kind of connections we can use between the funders. Things like Make Data Count may also help us as well. But also I want to think, we view taxonomists as people who find, discover, and describe species. I think they could be viewed more as biocurators. They're looking after physical specimens and the objects based on those. And if we extend that to also the things that are based on that, derived on that, images, specimens, and so on. So if we track those, and in particular track their citations, then we can learn a great deal about the impact of taxonomy. And if we can do that, then of course it would be trivial to answer this question, who does taxonomy, where, and who funds it? Thanks for your time.
And we do have a minute and a half if there are any questions.
Excuse me, how do you think about the recent change of the creation about zoological records? As you know that zoological records were published some paper version of zoological records, and some curation attempt of zoological records was very good, but recently the works by the zoological records is really bad. They provided the database, but usually this is not so comprehensive. Previously, it was done the very good work, creating a many, how to say, they focusing on the journal that don't have any impact factor or something like that. But recent database of the zoological records only focusing on the how to say major journal or something. So recently, that many taxonomists is very difficult to obtain the the information about recently described taxon or something like that. Sorry, my question is somewhat.
No, I think, so if I understood correctly, so zoological records has now become index of organism names, and it's published by Claveria, who run Web of Science, which Arturo was talking about. I guess part of the challenge is it's a huge job. The taxonomic literature is incredibly widely dispersed, and we have journals from very major ones like ZooTax or ZooKeys, right down to journals that literally somebody's publishing once a year. Maybe we should talk about it. I'd be interested to know if their coverage is declining and what's going on. But the challenge of indexing, particularly the zoological literature, is enormous.
Thank you very much.
And thank you so much, Rod.
Cheers.
Our next speaker is Arianna Salili-James. Thank you so much.
Hello, can you guys hear me? Okay, awesome. So hi, I'm Arianna Salili-James from the Natural History Museum in London, and I'm here to talk about robotics at the museum. So just for reference, I'm an AI researcher, not a robotics engineer, so if you do have robotics related questions, please be gentle on me. But yeah, I'm here to talk about the general side of the robotics project, but also some more AI related projects that we've got going with that. So my talk will be split into basic digitization ideas we have with the robotic arm, and also going into more detail about one AI project we've got in particular. So NHM digitization, I'm sure you all know a lot about that, but we've got a lot of data at the museum, and we've got an amazing digitization initiative going on. So we've got lots of specimens that have already been digitized, with over 10 years of NHM digitization initiatives happening. Now, amongst this, there are loads of really cool things that are going on. We've got lots of creative workflows and creative setups used for digitization. So one example is Alice, for example. I'm sure you've all heard of, well, some of you may have heard of Alice, but Alice was a setup designed to digitize the pinned insect specimens in particular. So it's useful for taking photographs of insects, but also of the labels on the pinned insect. Getting all those informations out from the labels that might not be seen from just taking a regular photograph using previous digitization setups. There's also lots of other things happening at our museum and at different museums and different institutions across the world, like scanning and robotics for book scanning and things like that. And this is something that we were starting to think about. So we asked the question, can robotics be integrated with museum digitization? So the aim here isn't obvious, it's not to replace any human digitizer with robotics, because that definitely cannot happen for various reasons, but can we integrate it with current setups? So can we use a collaborative robot or a cobot? So we brought ourselves a cobot. So just a side note, this is from a slide that my colleague gave last year at Hadwig, hence why there's a picture of me, so apologies for that, but you can see double me today, so yay. And we recently got a robot. We got a Techman TM5-900 that my colleague Ben Scott spoke about last year. So we're in the early stages. We've had it for a year and a bit, but we're still in the very early stages of this. So this is a robotic arm. It has a 5-megapixel camera. It also comes with a 3D camera, not with color though. So yeah, no color, just to give you a 3D structure view. Also has a reach of 900 millimeters, payload of 4 kilograms, and a built-in vision system, built-in computer vision system with pluggable and trainable AI. So that's kind of where I come into this. So 2 questions, sir. So the first question is, which collections can benefit from cobot integrated digitization pipelines? So what kind of collections can we be working with? Natural history collections? Are we talking insect collections or herbarium things? There's quite a lot we've got at the museum, so we have to have a little think about what we can do with this. And another thing is, how can we do this safely without destroying specimens and whilst keeping the curators happy as well? So safety is a very important thing, of course. So the robot does have safety precautions in there, so we can tell it how far to go, for example, not to move its arm too quickly, speed restrictions on that, and limiting it to a certain area. So that's something that we're thinking about. And other things are when we're doing this for training purposes, for example, training the AI, we want to either start with working on synthetic specimens or working with specimens that might not specimens that are used for training. So when I say training, I mean like when you have museum events and you want to show people some specimens, things like that we use for classical training things. And yeah. So previous projects, so my colleague Ben spoke about this last year, so I won't go into too much detail, but we started working on a variety of projects. So one was on shark teeth specimens. So shark teeth are less fragile than some other specimens. So we thought that was a good specimen to start working with. And a lot of the shark teeth at the museum are currently in boxes, and there's small boxes with huge amounts of shark teeth in them. So in order to digitize this, what the robotic arm would need to do is take the tooth out of the box, put it somewhere, take a photo from labial and lingual views, so take a photo and then turn the teeth around and then take another photo. And then put it maybe into some compartmentalized box instead of the original box. So that was the idea with the shark project. Again, very early days, baby steps we're taking at the moment. Another project we had going was using AI to do some species identification from above. So if your robotic arm is above a specimen drawer, can we classify species of insects from that view up there? And you might be asking why we're doing this, but one reason was depending on the species, the robotic arm might act differently. So if it was a butterfly, it might act differently to if it was a beetle or something. So those are just some things we were playing around with. So then we started to focus a little bit more, and we asked, can we use AI within cobot integrated pinned insect digitization pipelines, which is a much longer sentence than I intended, so I do apologize for that. So this was a project that I was working on with a student of mine at UCL, and the general aims are, obviously, can we use cobots for pinned insect digitization? So what does that mean? That means we want to photograph the specimen itself, but also we want to get all the data out of the labels on the specimen. Another more complicated thing is, do we also want to add identifier labels? So identifier labels are needed for digitization. So if the robot wanted to do that, then this is slightly more complicated. Right now, a digitizer will take an identifier label and potentially add it to the pin, which can be done with a digitizer, but we don't really want to trust a robotic arm to be pinning something like that. So we are thinking about other ways that can happen. For example, if you had potentially an identifier label with a slit inside it, then you can just attach the label like this, which is a much easier action for a robotic arm than to pin it. But of course, I think the easier option here would be to just have the digitizer do that step and the robotic arm doing the other steps. So what would this involve? So this will involve scanning drawers. It will involve picking and placing the specimen, so taking it from a drawer and putting it somewhere else to take photographs of that. And also taking photographs of the labels once you're doing that. And then putting it back again. Again, with the question of putting it back, there's the initial question of where do you put it back? Do you put it back into its original spot? Or if the robotic arm is adding the identifier label, then it can't necessarily go into its original spot because it's going to take up more space. And lastly, can we add an additional pipeline in here? So within the workflow, can we also potentially use the data that the robotic arm is getting to do things like 3D reconstruction using photogrammetry or more advanced AI methods for that? So yeah, pinned insect digitalization. So this is a workflow that my student from UCL, Neifeng Zhang, created. So it's essentially what I was just saying. We're using this to try and digitize the pinned insects, but we are working on the AI side. So we've yet to fully integrate it with the robotic arm. So purely dealing with the AI side at the moment. So the first step is to scan and detect the target. So if you've got a drawer of insects, the camera from the top can see all of this, and we need to be able to detect what is a specimen. So what is an insect, what is a pin, what is a label, and all of those make up what a specimen is. Once you've got that, then it will need to plan its route to go to a photography station. So by photography station, I just mean, in our case, just a bit of foam with a more plain background so you can take better quality photos. You don't want to take photos in its original spot because there's no room for the robotic arm to go if there are lots of other specimens there. And sorry, I just forgot one thing. Before you take it away, you need to know how to hold it. So we're working with pinned insects at the moment, so it's going to hold it by the pin. So when it zooms down, when the camera zooms in and the robotic arm comes down, it needs to work out how to hold that specimen, which part of the specimen to hold, and that will ideally be the top of the pin. And then lastly, when you do move it, can we find the labels around the specimen and detect the information on the labels? So this is not just scanning and segmenting labels, but it's also object tracking as well, because you want to know which label you're using. So we're working on all the AI stuff here. So the bits in green are the bits where we created models for. So the other thing is that we didn't want to use the robotic arm directly for this project, so we wanted to take our own data. So for training, we actually began using, again, the Alice-generated images that I spoke about, but also we wanted to mimic a robotic arm. So we have thousands of images or hundreds of images from Alice that we use, and we also took our own photos and took our own videos as well. So these were taken on a Canon DSLR and also on just a Samsung Galaxy phone and an older iPhone as well. So to mimic the arm, we would just go take a phone or a camera and go through the drawer slowly, like that. And then to mimic going up and down to detect those pins, we'll just move, again, the camera up and down. For the label tracking side, the label detection side, we wanted to show a pin specimen with a camera going around it. But given that that's quite tedious, instead we put the pinned specimen on a rotating platform, rotated the platform, and then the camera took videos of it like that. So specimen identification. So this is a nice video that my student Nathan made. So here it's going around and just essentially tracking the specimens from the drawer. So these were mainly trained on images, but they still worked really well on the videos. And the images were very, very high quality, but the videos were not high quality, but we still got really good results here. Lastly, it was trained on very different specimens. So it was actually trained on a bunch of dragonflies and caddisflies and beetles, but here it was tested on butterflies. So these were not seen at all during the training stage, but it still did a really good job at detecting what is a specimen. Pin locating is a lot more different. So when you work just on the insect, on the Alice-generated images, they are very, very high quality, and it does a very good job at finding the pin. And we're talking like 99% accuracy at minimum. But in real life, that's not the case. So when you work with videos, these are slightly more complicated. And also add the fact that we worked on different types of insects. So in our training set, for example, we just had we had some dragonflies and beetles, but we didn't have Lepidoptera in there. And some Lepidoptera come with fantastic black dots on them, which look identical to pinheads from top views. So at the beginning, the AI model was mistaking these black dots for pinheads. So we took we did a little cheat here. We then added a few images of butterflies into the training set. By a few, I literally mean a few. There were no more than 4 images we added. Once we did that, it really improved. It stopped thinking that a butterfly dot was a pinhead, so that was great. But then we had an additional problem, which was that sometimes there are multiple pins used for pinned insects. So you have the pin that holds the main part of the insect potentially, or the labels. That's the pin that we want to hold, so the one that has the labels. But there might be an additional pin holding down the specimen or holding additional parts of the specimen. And that is not what we want to pick up. If the robotic arm used that to pick it up, then it would be a disaster. So that was quite tricky. So we created an additional step in the AI model to detect which pin to use. And it actually worked quite well in that. And lastly, there's the label tracking. So identifying what is a label, that was fairly easy for an AI model to do. So that was great. Being able to track it was slightly more difficult, but still fairly doable from the few videos that we had. So again, this was mainly trained using just the images, but still worked well on the videos. Now, the idea here is we want to be able to extract the data from these labels. Now, we could try and do that automatically, so do automatic data extraction using OCR, using AI and various other computer vision things. But there is the added problem of what if the AI missed something out. So we still want to take a photo of these guys. But if you want to take a photo of the labels, then from which view do we do that? We can't just have a whole video of a label going on to the NHM data portal, for example. So what we thought about was, let's track these images of the label. So you have your video and you've got different frames. And once we track them, we can then group those labels, and then we can merge them together, related to all the other AI/Alice related work that my colleagues and myself have presented at TALB before. So please look at those papers for more references to that. Yeah, so that's worked quite well for us for now. So being able to use that to merge, to get all the label information out of those labels, sorry, of the pinned insects. So what next for us? Okay, we have 10 seconds left. So what's next is we're going to be using hopefully depth information to get the pinheads out, and we are also getting a new camera to get better quality images as well. Thank you.
Thank you so much, Arianna. We don't have time for questions right now, but you can find her during the conference week. At this point, I'd like to welcome up Mao-Ning Tuanmu, our next speaker.
Okay. Good afternoon, everyone. I'm Mao-Ning Tuanmu from Academia Sinica in Taiwan. So today I will introduce you a Soundscape data platform. But before that, let's listen to this. So we are listening to an audio recordings collected on a winter morning on a tea farm in Ali Mountain. So if we put time on the x-axis and the sound frequency on the y-axis and use different colors to indicate the sound intensity, we can visualize the recording using a spectrogram. From the recording you just heard, probably you might have recognized several bird species singing, and a car driving by and disturbing a great tree pine, and also some rain in the background. This is a great example of a soundscape, which is composed of all sounds produced by humans, non-human organisms, and non-biological processes at a location over a period of time. These sounds provide insights into the temporal patterns dynamics of the individual sound sources and also their interactions. So soundscape recordings are just like the nature history collections, providing the information about the spatial patterns and temporal dynamics of life forms across biological levels, and also provide the information about the interactions with the environment. With the low cost of data collection and the ease of scaling up, the Soundscape data can provide long-term and high temporal resolution information about multiple taxa. This makes Soundscape monitoring a crucial component of many biodiversity monitoring networks and a powerful tool for biodiversity conservation. So to preserve the acoustic records of the environment in Asia, the Asian Soundscape Monitoring Network was established in 2014. through a collaboration among researchers from Malaysia, Taiwan, Thailand, and Vietnam. To date, the network has collected over 22 million minutes of audible and ultrasonic sounds across more than 80 sites with various habitats. We use the two approaches to extract biodiversity information from the recordings. First, we use the two AI tools to detect and identify the animal sounds from the recordings. While the BirdNet developed by the Cornell Lab of Ornithology can identify more than 6,000 bird species globally, the CILIC developed by Taiwan Biodiversity Research Institute can detect more than 200 vertebrates, including birds, mammals, and frogs in Taiwan. Second, we calculated some acoustic indices to characterize the sound signals within the recordings. These indices can measure, for example, the frequency diversity of sounds in the recordings and the dominance of the non-human organism sounds compared to the human-made sounds. These data have made significant contributions to scientific research. For example, these data have been used to improve the information retrieval approaches, to characterize and understand animal behaviors and activity patterns, and to assess the impacts of environmental changes on biodiversity. However, we believe that the value of this data, including the audio recordings, the derived data, and the associated metadata, are valuable for many other applications. So to maximize the data value, we developed a data platform to mobilize the data and encourage their use. So this platform allows users to explore and access soundscape data for a variety of purposes. For example, if you want to explore the soundscape at a specific location, you can filter the monitoring sites by their habitat type and select one based on their geographical location. Once you click on a specific site, you can get detailed information about it. From the time series of 8 acoustic indices, you can explore the temporal patterns of different characteristics of the soundscape. Additionally, you can also easily identify some acoustic events from the long-term spectrogram, which is similar to the spectrogram, but over an extended period of time. If you want to know which species are present at that location, you can find a list of species which have been identified acoustically from the recordings collected there. You can also listen to the typical sounds of those species. Or if you want to access some recordings collected under certain conditions for further analysis, you can visit the data search page. There, you can filter the recordings by their metadata, such as the sound type, habitat, and the collection date and time. For each recording, you can view its spectrogram and listen to it to determine whether it meets your needs. You can then download the selected recordings, and each download will be assigned an ARK, which is similar to a DOI, providing a persistent identifier for easy citation and tracking of data use. However, to protect personal privacy, the recordings containing human voices cannot be played or accessed directly, although their derived data remain available to the users. So everything we have done is to ensure that the data are fair. A unique ID and the rich metadata associated with each recording enhance the findability of the data. All recordings collected within this network, except for those containing human voices, are accessible on this platform. And we are also working with the Soundscape communities in Taiwan to develop a data standard using the TechWix, the standard for the camera trap data as a basis. The use of the CC by NC license for all the accessible recordings, along with the platform's terms of use and the persistent identifiers, all enhanced the reusability of the data. With the fair data, we hope that more people will appreciate the Soundscape and its value, and also even can create new value by using those data in a variety of applications. So the launch of this platform is due to the hard work of the East Lab and the TIBIF teams, especially the co-authors of this presentation, Ms. Jiayun Li for the design and Ms. Jingying Li for the development. I also would like to thank our collaborators for their contributions to data collection and the founders of this project. Thank you all for your attention, and we invite you to explore the Soundscape with us through the Asian Soundscape Monitoring Network. Thank you.
We do have a few minutes for questions if anyone has a question.
Thank you very much for your presentation. Could you tell me about, do you have some special treatment for the sensitive data, like endangered species or something like that?
Yeah, currently we don't have that. Mm-hmm. Yeah, but I think, yeah, that's a good suggestion. So yeah, probably we will consider about that.
Okay, yeah. Thank you.
All right, let's give another round of applause and then welcome our next speaker. Our next speaker is Cailin Meyer.
Hi, everyone. Thanks for coming along today and sticking around for us. And I apologize in advance if I start coughing. I promise you I'm not sick. I just have a cough that refuses to leave me alone. There you go. All right, so in 2022, the Smithsonian Institution's National Museum of Natural History's Office of the Registrar entered a pilot program with the Department of Entomology to solve numerous legacy issues surrounding the Department of Entomology's acquisition and registration records and procedures. Many of the problems entomology is currently facing do relate to, say, the past 60 years of global international wildlife protection acts and how they have been historically interpreted over the several decades. While the language of these acts often implies for the inclusion of invertebrates, the historical interpretation has primarily focused on vertebrates and on marine life. My entomology colleagues have told me several anecdotes about attempting to apply for permits in early decades, only to be told by the officials that they didn't need them to collect insects. And this sort of scenario led to a long-standing culture where many of entomologists at NMNH have sort of assumed that permitting laws may not actually apply to their collecting. And it's only in the last few years that this perception has been slowly shifting in a manner that happens to coincide with all the increasingly stricter permitting laws in a post-Nagoya Protocol world. So the entomology department has also faced several internal challenges over the same time period. For instance, over 2 decades, we did not have a departmental registrar, and simultaneously, the museum was unable to provide any significant support due to similar understaffing and funding issues. And that had substantial impacts on our ability to maintain our registration records. Our technicians are hired and trained as scientists doing scientific research, but they were required to transact both loans and acquisitions, often without training or oversight from more knowledgeable staff. And this resulted in an interior culture where the technicians primarily relied on each other via word of mouth to solve very complicated problems. And many acquisitions went unresolved because they forgot to follow up with donors, or the donors never reached out back to them. It's a cycle, right? And there was also no standardization of record entry, as each technician sort of developed their own method of recording acquisitions. And I want to stress here that no one is at fault for this situation. This is not a case of anybody just deciding to not do their job. All of our staff were doing the best that they could with very limited resources they had available, in a complicated specialty that they were not trained in. And I'm sure that's a circumstance that we can all relate to. Entomology acquisitions also tend to be very large. So on this photo here, this is a stack of 12 entomology drawers that we've put together. Here they are, 4 stacks in 1 crate. And then in this photo, this is 9 crates worth of entomology drawers. There are over 40,000 specimens in these 9 crates. This is a single acquisition that we were given in 2019. This is 1 donation from 1 individual. And that's not an uncommon occurrence for us. Our average entomology acquisition involves over 100 specimens. And when I reviewed all the entomology records from 1990 to this year, I found that 20% of the acquisitions received contained more than 500 specimens, and nearly 10% of them contained between 1,000 and 10,000. And that also means that over a 35-year period, the department was handling an average of 35 of those large donations a year. So due to the previously mentioned lack of staff resources, and the fact that a lot of what we received happens to be unidentified or only identified to genus, it's impossible for the department to create really detailed acquisition records of each specimen that was involved in a single donation. We also lacked robust database technology to create those sort of records. The database systems we used prior to our current database EMU, they weren't as robust. They could not handle the same amount of data expected by modern best practices. Also, those best practice standards have in fact evolved because they are best practice standards and they should evolve. Many legacy records look like kind of skeletal records now, but they were acceptable at the time that they were written. And they often have descriptions as simple as 4,600 Heteroptera from the world. Again, that description leaves several things to be desired by today's standards, but it was perfectly acceptable in 1992 when it was written. So how do we start fixing this problem? In 2020, the OR approached the department about creating a pilot partnership. The OR would hire a dedicated registrar that would be permanently detailed to the department, and that person would focus on multiple registration tasks, such as resolving the acquisition backlog, providing the training and guidance to the staff, serving as a liaison of communications between everybody, and enforcing best practice standards for registration paperwork moving forward. So that dedicated person is me. Hi, hello. And on the screen here, I have a photo of what my office sort of looks like. And I'm sorry that registration work tends to be very boring because it's just paperwork, so I don't have fancy specimens really to show you. Also not shown in this photo are the other 10 cabinets of our holotype acquisition and catalog books, and also all the paperwork that I have inherited from the technicians. And the photo on the right is an example of one of the, I kid you not, 4 filing methods that I inherited. This one in particular stood out because it was alphabetical by donor's name, not through any particular year or anything, just alphabetical by name, including both first and last name, particularly at random. And it contained both resolved and unresolved acquisitions. Nobody in the museum today has any idea why. So I'm happy to say there's one filing method now. There's no longer 4, just one. So there are 2. At the start of this position, there were 1,700 legacy acquisitions that we had to resolve. And those are transactions that have been unresolved for 2 years, and they may lack a proof of an intent to donate, such as that data gift, or they lack a provenance information. Even though the acquisitions were obtained in previous decades, we're using best practice standards today to solve them. So that requires a lot of investigative work. And there's 2 key problems. The first is whether or not we have that data gift. The second is whether or not we have the permits, whether or not permits were needed, and what the permits actually say. So for those of us that do this work, we're not strangers to tried and true methods of hunting down donors, finding unsigned deeds of gift. I don't want to spend a lot of time talking about these tried methods, but I have to emphasize they do work. I have managed to track down legacy donor acquisitions via American public records for American donors. I have on one occasion cold mailed a new deed of gift to the last known address, and it successfully made its way to the donor, and they reached out to me via email. I kind of hate it when that works, right? Other techniques I have employed include researching the holotype. So if holotypes are mentioned in the inventory by genus species, there's a significant chance that the donor was the same individual who wrote that publication. And the type publications almost always name the repository that is now holding that holotype for study. And it also usually names how the researcher obtains it. So we can use that as proof of intent to donate. In instances where the donor is a researcher who's since moved to a new lab, looking for their last publication gives us a new address. And I've also discovered that many entomologists keep extensive records of their communications with collaborators, who in turn often become donors. These letters and emails hold evidence of somebody's intent to donate, such as the example here on the slide. And I was able to resolve 52 acquisitions this way, kind of proving that sometimes it really is better if people hold on to all of their paperwork, which sort of makes us sad. So the more prominent problem that we face is determining whether or not permits were needed. And I've sort of employed a Socrates questions method for this. First, what year were the specimens collected? The entomology collection has a unique problem where not all specimens were cataloged or tied to their acquisition when they were received. So it can be very challenging or impossible for us to completely unite them together. But knowing the donation year does give us a starting point to start evaluating the laws surrounding those acquisitions. And researching the donor's area of expertise and coordinating that with the recorded geographic area can help narrow down the countries where specimens were collected from. So in entomology, instead of writing down the country, it used to be very standard to just say the geographic area, and that was what mattered. But we can figure it out very slowly. It can also be useful to review the acquisition files for other donations within a similar time period to see if there's documentation for permits with those. And that can sometimes prove that permits were needed that we might be missing now. We can also ask our other departments if they have records of a similar time period that they can share with us if they happen to have those on file. There's also a significant number of resources available regarding historical environmental laws. My top 2 resources are CITES Lex, FOUL Lex, and EcoLex. These are essentially online repositories of legal documents from countries worldwide, and depending on your research, any one of them might have the legal documents that you are looking for a particular country. And I would also suggest turning to the humanities for help. I know. But the history of environmental law and wildlife protection acts is a very popular topic amongst legal historians, and we can often find papers that provide excellent background information, and they can also lead me to other resources. And finally, just because we don't have the permits now does not mean that we never had them or that they were not acquired. Many entomologists don't retain permits indefinitely when they enter our country, and those that do have lost them sometimes for completely valid reasons, such as a house fire because they happen to live in California. That is actually a case that happened this year. And in these instances, we obtain a written attestation that permits were granted and that the specimens were collected and imported legally. It's exceedingly difficult to prove a negative with absence of proof, and it's very difficult to say with 100% certainty if a permit was not required, if something is not so explicitly state it was not required. The best we can do is research and build a case that explains why we believe permits were or were not needed. Our directives and policies require us to have the legal ability to acquire and to have done our due diligence. And each acquisition is looked at on an individual case-by-case basis, and it's assessed within its own circumstances. And there may be instances where we have to refer the acquisition to the judgment of our Office of General Counsel. Those are our fancy lawyers. I am quickly running out of time, and I do want to leave some time for questions if need be, so I'll skip over this slide a little bit, but basically good communication is crucial to developing new relationships when there's a lack of trust amongst a department. It also goes both ways. And everybody has been very receptive to this new position, and we are all doing super great. So I want to end with a question to everyone here, which is, have you dealt with these similar issues surrounding specifically your entomology collections? What difficulties have you faced with them, and what methods or procedures have you found most beneficial with trying to solve them? If you've had similar issues, or if you're dealing with similar issues now, we would love to hear from you. Please let us hear from you. And on that note, thank you all for listening to me, and I look forward to a super exciting Spinach Tagwood 2024.
We do have time for one question if there's a quick one.
Hi, just as you put a lot of effort obviously into finding these donors. Once you've got biographical information on them, where do you put it? Do you just put it in your collection management system? Do you push it out anywhere?
No, we do not push it out anywhere. It remains within our system, kind of closed. It's not publicly available information.
Okay. The reason I say this is because a lot of those donors will actually be collectors, and I'm very interested in obtaining data on collectors of specimens in order to link them to their specimens.
Oh, I see, yeah.
Yeah. Obviously, you'll have privacy concerns
exactly,
if they
yes.
are alive, but if they're dead, any chance of you sharing them?
I'm afraid I'm probably not the person to ask that question to, but I'll be happy to refer it to my boss who will reach out
please
to you.
do. I'm very curious about that type of data. Thank you.
Love it. Great question. Okay, well, we're going to transition to our final speaker in this session, and that is Aaron Cooperwitz.
All right, can everyone hear me okay? Sweet. Thank you so much, everybody, for joining us. I'm bringing up the rear here, the last talk of the evening before the big reception. So I'll try to keep it short and sweet, but also leave time for questions, okay? I'll be talking about big charismatic mammals in a taxidermy collection at the Connecticut State Museum of Natural History, based at the University of Connecticut, where I'm a curator and collections manager, and I'm also affiliated with the Department of Ecology and Evolutionary Biology, where I manage the research vertebrate collections. All right, so it's pretty well known, especially in mammal and bird collections, that there's a sex bias in collections. And this sex bias for certain groups of animals can equate to a size bias. So that's what I'll be looking at a little bit today, especially for species or groups of mammals that exhibit SSD, or sexual size dimorphism. I'll be talking a lot today about Artiodactyla and Carnivora, orders where males are larger and more ornamented than females. A lot of this work has been done only in research collections, so collections where there's a lot of a priori specimen measurements taken. Taxidermy, that's not usually the case, especially with our collections. And so I wanted to kind of delve into, do we see a sex bias, but particularly a size bias in our taxidermy collections, using one particular collection example. So we all know that taxidermy in natural history museums is very foundational to the modern museum experience. We want to engage public, and we want to inspire awe and wonder in the natural world. That's kind of one of our big goals as educators and researchers. But we might also inadvertently be biasing public perception if we're displaying particularly large, ornamented, and kind of extravagant animals that might not exist in nature anymore. A lot of collections, taxidermy collections, come about through donations from trophy hunters, especially animals that were collected in kind of the mid-1900s, kind of 1950s, 1970s. And so I had a question that came about while we were appraising our taxidermy specimens in the CSMNH was, do extremely large mammals still exist out there? Are we seeing really rapid changes in body size that could be anthropogenic effects of hunting on natural populations? So what do we see today? So again, very preliminary study that I'll be presenting today, kind of a case study using only one collection. But I really was interested in learning how different CSMNH mammal taxidermy, again, which was collected in most of the 1950s, how the body sizes of these large mammals are different when compared to their species in contemporary populations. So focal group today is the Henry S. Budney collection, again in the CSMNH, donated in 2006 by one of his sons. These were all collected in Africa, North America, primarily a few neotropical organisms as well, but hunted out in the 1950s and 70s. The collection comprises full mounts, shoulder mounts, rugs, skulls, and antlers. I'm only going to be presenting full mount data today. So Henry Budney, again, a trophy hunter, he kept really good records on a couple of his expeditions. This was where he was going out to Mexico and hunting the tigre, the jaguar. And we even have some photos that we were able to find in the collections. However, despite all of this, we have very poor, in fact, non-existent body size data, which is really unfortunate. So we had to take the measurements now using mounts. The measurements that I took with help of one of my students were HB, or head body length. This is from the tip of the nose of the mammal to the inflection point of the tail. And I also took tail length, abbreviated T, which is from that inflection point of the tail with the body to the tip of the flesh, not including the little hair tuft. So think of like a lion's tail, it has a little tuft on the end. We didn't consider that, just where the tip of the tail of the skin ends on the tail. And we went into the collections and we measured them. So actually, this was really logistically difficult. We have a lot of our mammals stored in kind of a cramped space. I'm sure that's kind of a common theme with a lot of collections. And just kind of like doing yoga poses to get around and try to like measure them and get up on ladders to try to get at some of these big mammals. So again, head body length and tail length were what we measured. A pretty small sample size. I'm going to not be showing any kind of analyses, just some trends that we can discuss kind of with the question session too. But we ended up measuring 23 artiodactyls, so these are hooved mammals, indicated by those little silhouettes there. And then we had 11 carnivores that we were able to measure. And you'll see a few little duplicates there. So these are individual specimens, and we had a couple repeats of species, but usually there was just like one species, one specimen, so not a lot of power. And comparing our taxidermy specimens, I wanted to look at the most up-to-date contemporary populations of their species counterparts that were available. I mostly used the mammal base and the amniote database to get kind of the most current head, body, and tail length data that's out there, but I supplemented this also with Walker's Mammals of the World when I couldn't find really current information. I'm going to show you an equation, so don't be too scared here, it's very simple. The calculations I used to measure head, body, and tail length, I took the data from the specimen in the museum, subtracted the data that I gleaned from those databases related to the contemporary population, and got a difference in centimeters. And I want to highlight that I'm reporting things in centimeters. It's usually convention to report things in millimeters. I just think it's a little more interesting and maybe easier to wrap our minds around centimeters instead of millimeters. I'm first going to present tail length, which kind of had an interesting pattern in the things that I found. And I'm going to describe this figure. This will be kind of the same type of figure that I'll be showing you for the head body length as well. So on the y-axis, the vertical axis, you can see the difference in tail length in centimeters, again, reported on that central space, that's the 0 line. So if there was absolutely no difference, remember we're taking the value from the taxidermy specimen in the museum, subtracting that contemporary value and getting the difference. So if there was no difference, there was no bar on that 0 line. The blue bars showed the outcome of that museum specimen being larger than the animals in contemporary populations, and the orange bars meant that the museum specimens were smaller than the contemporary populations. Are we all good? Cool. Each of the little silhouettes indicates an individual specimen that was measured, and an interesting and kind of unexpected trend, and again, this is tail length, so take it as you will. Most of the CSMNH mounts had shorter tails than their contemporary counterparts. I don't really know how to explain it. I'll try a little bit in the discussion section at the end, but that was an unexpected result. I tried to then kind of look at the information and break it down taxonomically. So looking at the carnivores versus the artiodactyls to see if there was any kind of trend. Was one kind of larger or smaller or not? And looking at the carnivores, so those are in black, and I've kind of grayed out the artiodactyls, almost all of the carnivore taxidermy had shorter tails than the current populations. Looking at the artiodactyls, some specimens had much longer tails, but again, not a real consistent pattern. Things were kind of all over the place. The only thing that I found here was that most of our specimens had shorter tails, whatever that means. Now, moving on to the head body length value, which I think is a little more interesting and really tells you more about size and again, overall body size of these specimens compared to their contemporary counterparts. Same setup here. The scale on the y-axis is a little bit different, but overall, most of the CSMNH mounts had larger bodies than contemporary counterparts. And that's kind of what we had expected going into this, thinking that hunters had removed the exceptionally large mammals from the population. They didn't proliferate and breed, and we're not seeing really big things these days compared to like 70 plus years ago. So, but it wasn't a huge disparity. So 19 out of 33 of the mounts, considering both of those groups, did have larger bodies. Breaking it down taxonomically again, so looking at the carnivores first, there really wasn't a consistent pattern. And I think some of these, like that bear that was like 40 centimeters smaller than a contemporary counterpart, I don't think that was an adult. And these were also adult male values that I was looking at for all of these specimens. So there's a few things there that maybe we would take those out of the data set in the future, but carnivores did not show any consistent pattern. But the artiodactyls were the largest specimens in the museum, which kind of went along again with what our observations were. So now I'm zooming in on just the larger specimens, kind of forgetting about the ones that were a little bit smaller than the contemporary counterparts, and again, focusing on that head body length value. The 10 largest taxidermy mounts in the collections ranged from 10 to 38 centimeters larger or longer than their contemporary counterparts. So that's pretty big. And that big one there is our good friend, the moose. And actually that individual was the one that the appraisers pointed out as like that specimen. You will never see animals that big in the wild anymore. Again, because it's presumed that they've been hunted out and they couldn't proliferate. So almost a 40 centimeter difference in the size of our large specimen compared to moose that you would see out in the field today. Was kind of interesting. Again, this is still really preliminary, but I think there's a lot of cool directions this could go. So kind of digesting that a little bit, I know I'm zooming through this for you all, but I want to kind of distill everything down into a short take-home message. I mentioned I didn't really know what's going on with that tail length shortness, that overwhelmingly most of our taxidermy mounts had short tails compared to extant populations. I don't know if this is just a byproduct of the mounting method, putting a little wire through the tail, and the skin kind of tends to shrink more around that, maybe than a body form that had been sculpted. I don't really know, and I totally welcome suggestions as to why you think this might be. Moving on to that head body length largeness, especially for the hooved mammals or the artiodactyls in the CSMNH collections, the largest mammals had been removed from populations by hunters. Again, most of our specimens in this collection were hunted out by Budney and hunted in the mid-1900s, in the 1950s to the 70s. So there is actually, maybe we're seeing this really rapid effect of smaller mammals appearing in contemporary populations. So in about 70 years, we can see these traits changing. I don't know if this is a true pattern. Again, no analyses affiliated with the data that I presented, but I'm very curious to see if this holds in other collections as well. And this is kind of highlighting something that we're hoping at the museum to kind of capitalize on in the near future, to look at anthropogenic effects on big mammals. So we want to have a big public display and kind of take this as an opportunity to highlight, maybe we have things that are a little bit larger than what you would see nowadays, and what that means, highlighting how humans have affected and probably will continue to affect big mammals into the future. I would like to thank especially Lily Kane, an undergraduate student who was working with me to climb ladders and take all of these measurements in our storage room, along with the museum director and my friends. And with that, I would like to take any questions. Thanks very much.
So I've got a couple of online questions.
Oh, cool.
One which might be easy. Do you know the locality data for the large moose?
No, I don't, unfortunately.
And then the second question, did you account for specimen preparation methods such as the use of pre-made form or the tanning method and stretching shrinkage of the skin?
Yeah, I didn't really go too into detail with that, mostly because we don't have that information. Again, it's a real lack of data on these guys. We're assuming kind of in that era, and Budney, again through anecdotal information, was very cognizant of trying to find taxidermists who could prepare these things accurately. And so we're just making a whole load of assumptions here, that the forms that the skins were stretched over did match what these animals looked like in life, in reality. So we hear that he did aim for accuracy, but again, lots of assumptions with this one, yeah.
Hello, my name is Dmitry. My question is, you mentioned natural population of animals. Did you compare those with zoo animals, which tends to live longer and presumably became larger?
No, so those weren't, it wasn't explicitly stated in those trait databases that I was gathering the contemporary information from, if they also incorporated zoo animals, but that's a really interesting avenue to also look at. But no, they were kind of like broad umbrella term. This is the head body length for this species. I don't even know how, it's average values, and I don't know what the average values were taken from. But yeah, good point.
Hello.
Hi.
Oh, yeah. Hello.
Yep.
My name is Cecilia. I was wondering if you have thought about looking into fish data. Like, I guess you could see some pretty big changes in there. I see mammals are much more fun for the audience, but maybe that could be a good story to tell as well.
Very much so, and I think that's going back to like that sex bias literature. I think, again, birds and mammals were kind of the first groups, and again, research grade specimens there. But I know that there are some fish literature out there and some herp literature as well, looking at more from the sex bias side of things, but then seeing if that could equate into a size bias. At the museum, we don't have, I think we have like 3 fish taxidermy, so that wasn't something I could explore with our collection, but other collections could definitely.
Cool. Well, thank you so much, Erin.
Yeah, thanks.
And this is the close of today's conference activities, except for the reception, which will be at the Laguna Hotel starting at around 6:37. So the Laguna Hotel is just a short walk that way. And then for tomorrow's conference activities, just a reminder, as Dave said earlier, those will not be here. They will be in room B, area B. There will be signage apparently, and it's also that way. So we'll see you tonight for the reception in about an hour, and tomorrow at 9 a.m. And thank you all so much.